All right. Well, thank you for the chance to present. Thank you for the introduction, Hal. My name is Michael Herriges. I'm a postdoc in the Cotton Lab at BU. And today I'll be talking about my work on transplantation of mouse pluripotent stem cell-derived tip-like cells into immunocompetent mice. So as most of you already know, in the case of severe pulmonary disease, one treatment option is lung transplantation. But this is heavily limited by the supply of donor tissue and the eventual immune rejection of any transplanted tissue. So there's a need for other techniques to replace or replenish the lost respiratory tissue. One hypothetical technique, as Hal kind of mentioned, is iPSC-based cell therapy for pulmonary disease. So in this case, we have a patient with pulmonary disease. We can collect their cells and reprogram from the blood, reprogram into iPSCs, use CRISPRs to correct any disease-causing mutations. Then, using current or future techniques, we can differentiate these cells into various lung cell lineages with the hope of transplanting these cells back into the patient to replace any cells lost over the course of disease. The advantages here, we don't have to wait for the full tissue to be available. And since these cells come from the patient originally, they shouldn't be recognized as foreign, and there won't be a need for immunosuppression. Now, this sort of treatment is obviously many, many years away from being a reality. Really only in the past 10 years have we seen papers we like that have shown transplantation of any cell into mouse lungs, let alone human. As an example of this, there's a 2017 paper where the authors developed a lung progenitor media, or LPM, which could be used to culture embryonic lung progenitor cells known as TIP cells. And in this media, these epithelial cells maintained their progenitor state, and they could then be transplanted into injured, immunocompromised mouse lungs, where after 2 months, they saw these cells giving rise to cells expressing alveolar type 1 and type 2 cell markers. So this is really promising, but again, it's dependent on collection of embryonic tissue, which limits translation into clinical therapy. So this is where my project came in. We wanted to take a step from here, closer to that eventual human iPSC transplantation goal by asking, can we generate a mouse embryonic stem cell derived cell population similar to these transplantable primary tip-like cells, with the ultimate goal being mouse into mouse transplantation. Towards this end, we developed a protocol building off work in our lab and others. We're using a ES cell line with a NKX 2.1 M-cherry reporter to mark lung epithelial cells. We differentiate through anterior foregut endoderm into an early lung epithelial state, at which point we can sort purify our progenitor cells and grow them in the same LPM cell culturing condition to create what I'll be calling ESC-derived tip-like cells. These cells grow out in monolayer epithelial spheres, as shown here on the left. They look very similar to the primary counterparts. Both the primary and ES-derived cells can be passaged multiple times where they maintain their proliferative state. They can be frozen down and thawed at a later time. And importantly, our ES-derived cells maintain expression of this NKX reporter throughout this passaging. I don't have time to go into it today, but we characterized our cells by single-cell sequencing and qPCR, and at least on a transcriptional level, our ES-derived cells are very similar to the primary counterparts. So given this transcriptional similarity, we next wanted to see, can we transplant our cells similar to the primary tip cells? To do this, we labeled our donor cells with a lentiviral GFP. We then injured some syngeneic wild-type mice with bleomycin, and 10 days later transplanted our cells into these same mice. If we follow them out 6 weeks post-transplantation, we're happy to see cells expressing M-cherry and our lentiviral GFP, indicating the presence of donor-derived cells. Importantly, since this is a NKX 2.1 reporter, we would expect these cells to express this important lung transcription factor. And indeed, the vast majority of our donor-derived cells express this protein. We can then zoom in on some of our GFP-labeled cells and start staining for cell type-specific markers. In the white arrows, we see cuboidal cells with punctate surfactant protein C, characteristic of alveolar type 2 cells. And then with the yellow arrowheads, we have thinner cells with protoplanin expression, characteristic of alveolar type 1 cells. So this is at 6 weeks, a time point that's been used in previous publications, but we really want to see if our cells are lasting for a long time in the mice. So we followed some mice out for 6 months post-transplantation, and even at this time point, we can still see the existence of GFP positive and mCherry positive cells, again expressing NKX 2.1. And again, as at 6 weeks, we can zoom in and see cells still expressing that punctate surfactant protein C, as indicated by the white arrows, and protoplanin positive type 1-like cells, as indicated by the yellow arrowheads. So again, we seem to be getting type 1 and type 2-like cells as late as 6 months post-transplantation. But these are only a couple of markers. We really want to get a more thorough interpretation of what our cells are. So to do this, we turn to single-cell RNA sequencing. Here again, we're transplanting cells 10 days post-bleomycin injury, and then following out for either 6 or 15 weeks post-transplantation. At 6 weeks, we're looking at an uninjured control where we have no transplant, no injury, just take some of the endogenous cells. And then from one of our transplant recipients, we take all cells that express either mCherry or GFP, as well as the fluorescent negative, presumably endogenous cells. At week 15, we took a wider approach, just in case our donor-derived cells were giving rise to any non-epithelial lineages. So here we took all cells that were mCherry or GFP positive, and then as well, again, an endogenous control of just EBCAM positive cells. With a lot of bioinformatic processing and analysis, we generated this spring plot where our endogenous cells are in purple, blue, and green, and the donor-derived cells are in yellow and red. Importantly, this plot is only showing our epithelial cell types. This was because we didn't see any donor-derived cells in the non-epithelial lineages. We got over 15,000 cells here, the vast majority of them expressing NKX 2.1. And while our cells were highly proliferative prior to transplantation, by this time point, we see minimal expression of proliferation markers, suggesting we're not getting any sort of tumor forming. So as you can see here, we've got multiple clusters of cells suggesting different cell types. To tease apart what these are, we generated some cell type gene signatures. So these are each 20 to 35 genes enriched in the various epithelial cell types. Starting with our alveolar type 2 cells, we have an endogenous cluster with high expression of the signature, but also a donor-derived cell population with more moderate expression of these genes. Likewise, type 1 cells, we have a streak of endogenous type 1 cells, as well as a streak of donor-derived type 1 cells. In contrast, if we look at various airway markers, such as secretory, ciliate, basal, or neuroendocrine cells, these are not expressed in our donor-derived populations. So based on these signatures and Louvain clustering, we labeled our various cells with the various lung epithelial cell types. I do want to just call attention to this last population of gastric-like cells. This was a rare population of our week 6 donor-derived cells that don't express NKX 2.1, but instead have some of these gastric markers. While this was kind of unfortunate, it lines up with previous publications showing that loss of NKX 2.1 in the lung epithelium can lead to this sort of gastric phenotype. So not great, but lines up with some previous endogenous phenomena. But again, the vast majority of our cells are these type 2 and type 1-like cells that are close but not overlapping with their endogenous counterpart. So we wanted to see what genes are differentiating our cell populations here. And here we just have a heat map looking at the most differentially regulated genes between our endogenous cells in the yellow column and our donor-derived cells in the green columns. And what we saw for type 2 cells is our donor-derived cells had lower expression of some maturation markers, such as SLC34A2, LAMP3, LIS2, SPB, but had an even more striking loss of a lot of major histocompatibility complex 2 proteins, such as CD74 and all of these H2 proteins. Similarly, our type 1-like cells had lower expression of some maturation markers like protoplanin, VEGF-A, aquaporin-5, but again had this deficit of MHC2 proteins. So in total, we're getting this maturation lagging slightly behind, and this deficiency in MHC2 genes makes some sense. Our cells are coming from a very sterile environment where we know they don't express these genes. And so it seems that even after 15 weeks in vivo, the cells aren't turning these genes on to endogenous levels. But this is, again, focusing on what genes are different. We also wanted to take kind of a more broad approach. So here we're looking at Pearson correlation, looking at the 500 most variable genes in all of our single cell data. Here we're breaking up our various cell types by their original sample. So for example, in purple are our type 2 cells with the endogenous and donor-derived cells split up. And we're correlating each population against each other population, with the diagonal being comparing a cell population against itself, perfect correlation giving you this big red circle. And I just want to call your attention to these two triangles of high correlation, and these correspond to comparing our type 2 cells against each other and comparing our various type 1 cells against each other. So even though our donor-derived cells are lagging behind in some maturation markers, on a wider scale, they're still highly correlated with their endogenous counterparts. With that, just to summarize, we generated a protocol to make ESC-derived lung progenitor cells that are transcriptionally similar to their primary counterparts. These cells can be transplanted into immunocompetent recipients where they survive for at least 6 months and give primarily rise to alveolar type 1 and type 2-like cells. When we look closer, these donor-derived cells differ from their endogenous counterparts due to lower expression of some maturation markers and MHC2 proteins. Moving forward, we really want to get towards functionality. Do our donor-derived cells form lamellar bodies as endogenous alveolar type 2 cells do? Also, we want to see, can our donor-derived cells proliferate in response to a second injury? So we've started up some experiments where we do a transplantation and then a pneumonectomy to hopefully trigger proliferation in our cells, which we'll be planning to assess by EDU. And then in the very long term, we want to do transplantations into various disease models to try and rescue those mice. And then, of course, keep trying to improve our cells, make better donor populations, and better understand what is happening to these cells after the transplantation to kind of mature them into the state we see them. And with that, thank you to everyone, especially the Cotton Lab and Daryl in particular. Thank you to all our collaborators and support, and happy to answer any questions. Thank you.